Hello, class. Today we're going to talk about chapter 4 in the textbook, how neurons use electrical activity and electrical information to communicate. Next week, we'll talk about the chemical signaling that these neurons do at the same time through neurotransmitters, and that will bring us to the end of the material that will be on the first midterm. Everyone's doing quite well on the quizzes. Keep up the good work. So we're going to go through these 4 sections today. We're going to think about how throughout history, scientists identified electrical activity in the brain and the tools that we use to study it. We're going to talk about the bilipid membrane between the interior and the exterior of the cell and how there's electrical potential across it. And then we'll talk about how neurons combine information from multiple other neurons to create an output. And then we'll finish today's lecture talking about how the input to the nervous system and the output to our muscles. This picture goes back to the time of Descartes, in which Descartes proposed that one way that the nervous system might transmit information is through fluids. So he thought that there might be some long tubes in our body filled with fluids that might be impinged upon by things like heat in the environment, which would move through the tubes and create a chain of events that ultimately would lead to our muscles moving. This turned out not to be correct, but we can see in this theory the parts of a theory that we would need to explain our brain and behavior. We need to explain how nerves detect the sensory world around us and how they transmit this information they detect to the brain. We need to know how the brain decides from all this input what to do. And we need to know how the brain sends signals out to the muscles to produce a response. Descartes had this time proposed that the fluid would expand the muscles and allow them to move. And we'll see today that it's a little more complicated than that and involves electrical chemical signaling at the muscles. Before we get into the nervous system, we'll talk a little bit about electricity and electrical potential, just as a refresh. Recall that electricity is a potential for energy to occur. And so, for instance, in a battery, there's electrons that have a potential to travel from the negative to the positive side of the battery. Because the more electrons are on the negative side, we call this the negative because they have a negative charge and they're attracted to this positive side. So if we close this switch, the electrons will flow and light up this light bulb. And so electrical potential, which we'll talk about today in this case, is the ability for work to occur. The ability for this energy to move from here to here. So a battery has electrical potential because it has this buildup of negative ions that want to move to a positive location. So electricity is a flow of electrons from a place where there's more to a place where there's fewer. The negative pole is the source of those electrons. And we say it has a higher charge, and the positive pole is where they're flowing to, or the sink, and we say it has a lower charge. In the 18th century, it was beginning to be understood that our nervous system might have influence from electricity. People were dissecting nerves and found that electrical current being applied to them allowed the muscles to twitch. So Galvani proposed that electricity flows along the nerve. So this is a first indication of something called electrical stimulation, where you can use electricity to stimulate the nervous system to try to modify the activity of the brain cells there. So because the brain uses electricity to communicate, you can change this communication using these stimulation techniques. Some of the first stimulation studies were done in the 1800s. First by Fritsch and Hitzig, who were stimulating the neocortex of animals and had the arms and legs of the animals moving. And a gentleman in America named Bartholow, who implanted an electrode into the brain of one of his patients who had an exposed brain from a hole in their skull. And when he passed current through that electrode, the muscles of the body of the patient moved. So if we isolate a single neuron, a nerve, for example, in a petri dish, like in these diagrams, we could think about both electrically stimulating that nerve or recording the activity of that nerve with different equipment. So we could use a stimulator where small millivolts of electricity are passed, like the closing of a battery. So a little switch of that battery will close and pass current from here to here. And this will change the electrical dynamics of the axon without damaging it. You can also record electrodes, record from neurons this way, so you can measure the difference in voltage between two locations on an axon, for instance, by putting your needle and your recording electrode on two different parts of the axon and measuring the voltage potential between them, just like you may have used a voltmeter to measure the how much voltage is left, how much energy is left in a battery. Okay, so from this idea comes these new technologies like EEG, where we can stick electrodes right on the surface of a human's brain. We're measuring those same electrical signals, just clouded by this skull and scalp. So Catan in the early 19th century was trying to measure the currents of the brain in this way using a voltmeter and electrodes attached to the skull, which was therefore one of the first examples of the electroencephalogram, or EEG. So this is the voltage over time between two locations on the scalp. And these represent, as we'll see, these things called graded potentials of many, many neurons all together. Graded potentials are the input to a neuron. Von Helmholtz in the 19th century realized that because of the speed by which the information was moving down axons was too slow, that it couldn't possibly be electricity that was moving, like in our wires in our offices, where it's moving at the speed of light. He proposed that instead it was moving much slower. And so it was thought of maybe that it's not actually the electrons that are moving, but a wave of information moving down the axon, which we'll see turned out to be true. So as just to cover this again, an electrical potential is a charge. It's measured in volts or millivolts or microvolts. It's a potential energy, ability to do work through the storage of energy. A volt is a measure of a difference in potential between two locations, and a voltmeter is a device that measures that difference in potential. And an EEG is an example of a complicated voltmeter. So one way we can visualize a difference in potential between two locations in a body, for instance, is one cavity might have many more electrons and one might have many fewer electrons. And so we can think about, or even in a battery as well, this might be the negative or positive side of a battery. We can think about the electrons wanting to flow, the current wanting to flow from the negative to the positive side. And so this difference between negative and positive is what we call electrical potential. And so when incoming signals stimulate the cell body, they change the voltage of the neuron to a point where it sends a change in voltage down its axon, which we can measure with voltmeters as we go. So if you have a voltmeter in a particular place on the axon, every time you stimulate the neuron and put an output into it, you'll be able to see at a certain time the voltage change along that axon. It was only when scientists identified creatures with very, very large axons that were able to be measured by their rudimentary equipment that they had at the time that they could make a better understanding of how the system worked. So they identified a giant axon of a common squid in the Atlantic Ocean. The axon had a much larger diameter than our axons, which are in micrometers. This was almost a millimeter wide, and so thicker than a human hair. And this was the main animal that was used in the Hodgkin and Huxley experiments in the 1930s and 40s in America, which went on to win them the Nobel Prize. Hodgkin and Huxley came to understand how information travels down these axons and understood the action potential using this animal model. So they dissected the giant axon out of the squid and they put it in a salty bath in a petri dish. And then they used electrodes and voltmeters and oscilloscopes to measure the activity of those axons. So an oscilloscope, as I alluded to, is a very sensitive voltmeter that can record the voltage over time into a graph. So when you see someone's EEG in the movies, you're seeing an oscilloscope measuring that voltage over time. So we can display them on a graph in real time, or we can load them into a computer and record them, which is often what we're doing today. So the horizontal axis of these graphs is going to be time, and the vertical axis is going to be voltage. And here in this graph, we can see two action potentials recorded from a neuron, where the voltage changes very quickly from negative to positive. And we'll get back to this, but by convention, we say that the resting membrane potential, the difference between the inside and the outside of the cell is minus 70 microvolts. So just like a battery, the neuron has a potential energy, has more charge, negative charge on the inside than the outside, and the membrane separating the two, keeping that charge from moving. So one other thing we need to measure electrical activity from these neurons is something called microelectrodes. So these are electrodes that are small enough that they can fit inside or next to an axon and record from a single axon. So they can measure a neuron's electrical activity, and they can also stimulate neurons. There's two types of microelectrodes that scientists can use. One would be a metal wire, which has no insulation at a very fine tip, and then the rest is insulated. This gets recorded onto the oscilloscope or voltmeter. Alternatively, you can use a small glass pipette filled with salt water connected to a wire. And this pipette is a useful tool which can be used to suck up a little part of the membrane due to something called a patch clamp. This can isolate that tiny part of the membrane so that the recordings are predominantly from the receptors in that tiny part of the membrane. Okay, so you place the tip on the surface of an axon, and you put the second electrode inside the axon, and you can therefore measure the voltage across from inside to outside the membrane, which we'll see is about minus 70 microvolts. And we talked about this patch clamp where you suck a little bit of the membrane inside the pipette so that you're only recording from that small section of the membrane. This is how scientists can understand things like gates and channels, by isolating them in this small space. Just to cover this again, cations are positively charged ions like sodium and potassium, and anions are negatively charged ions like chloride. Turns out that all the proteins we talked about last week that are created by our RNA in our endoplasmic reticulum and sent out by our Golgi apparatus into our cells all tend to have a negative charge as well. So this negative charges of the protein molecules contributes to the negativity on the inside of the cell. So all these chloride and sodium and potassium ions floating around inside the cells, some on the inside, some on the outside, can create potential for movement. And there's a couple ways this can happen. One is through simple diffusion. So if you have a very high concentration thing like a drop of food coloring and you drop it in a tank of water, that food coloring will diffuse randomly throughout the water until it's equally distributed throughout the water. And this force occurs through random motion. So it's not necessarily an active force, but just a spreading out through randomness. Alternatively, or relatedly, the concentration gradient, therefore, is the difference in concentration between two regions. And so there's potential for one particular molecule to travel from one area to another if there's more concentration in the first area than the second. So that's why it's called a gradient. You can think about it like a slide, like there's a force pulling a molecule from an area where it's densely concentrated together to an area where it would be more spread out. Alternatively, there's something called a voltage gradient, which we've talked about with respect to a battery already. It's a difference in charge between two regions, and it allows a flow of current between the two regions if they're connected. So ions want to move from an area where there's higher charge, there's more electrons, to an area where there's lower charge or less electrons. So they want to move from an area like the inside of the cell where it's very negative to the outside of the cell where things are more positive. So here's an example of the concentration gradient, a little bit of ink distributing equally throughout the water, and a voltage gradient. If you pour salt water into water, all those positive and negative charges will repel each other perfectly until they're equally distributed throughout the water. So we can think of a cell membrane as a dam in this operation that only lets some of these ions through and only at certain times. So for instance, if you place salt in one side of a beaker with a cell membrane between it, and you have an empty bunch of water in the other side of the beaker, at first the salt will be very, very concentrated in a few crystals, and then through diffusion it will completely dissolve into that section of the water, but it will still be kept from moving to the other side of the beaker by this membrane, which is acting like a resistor or a dam, resisting the concentration gradient. And so not only does the water want to move, what does the salty water want to move to the freshwater side because of the increase in salt, but also because the increase in charge. So there's a large degree of potential energy here across this membrane. And we can think about this like a battery. In fact, this is almost the same setup as most of our batteries. They'll have a bunch of negative ions and positive ions separated by an insulator. So in reality, though, this membrane has proteins that cross it. And these form little channels or gates or pumps, as we saw last week, and they allow certain things to pass through. So for instance, if these were chloride channels, then chloride ions could pass through because they're more concentrated on the salty side than the less salty side. And the negative charge also will pull those same chloride ions over. But because they're also attracted to the positive charge, they won't all move over. So there'll be kind of a halfway point where enough will move over so that there's almost equal amounts of chloride on either side. But some are still being attracted to all the positivity on the other side. And you can think of this kind of dance ending in something called equilibrium, where the forces pulling them to one side and pulling them to the other side have balanced out. So that half the container will have the same charge as the other half, and half the container will have the same concentration as the other half, unless those are imbalanced, and then those imbalances will cancel each other out as well. So you can see here how this mix of a voltage and a concentration gradient can create interesting distributions of positive and negative ions on either side of cell membranes. So the chloride ions wanting to move with their concentration gradient into the less salty side is counteracted by chloride ions wanting to come to the positive side because they're negative. And an equilibrium will occur when the concentration gradient on the right side of the beaker is balanced by the voltage gradient on the left side, when they're perfectly balanced. So the concentration gradient will come to be equal to the voltage gradient, and they'll oppose each other perfectly. And Hodgkin and Hogsley actually formalized this in a set of equations, which you can learn about if you're inclined. So when we do this at rest and we measure the electrical potential between the inside and the outside of the cell, and we don't stimulate the neuron at all, we see that the neuron is storing negative energy on the inside compared to the outside. And if we say that the outside is 0 as a reference, then the inside of the cell is usually around -50 to -70 millivolts more negative than the outside. So we can know this by measuring with an microelectrode on the inside and outside of the cell. And we see that this constant uninterrupted voltage of around -70 microvolts from the inside to the outside of the cell. So this potential energy, this -70 microvolts, is a stored energy between the inside and the outside that can be used when certain channels or gates are opened up. Now, the resting potential of the neuron is created by the sum of these 4 molecules: sodium and chloride, salt, which are in higher proportions on the outside of the cell, and potassium and these large protein anions on the inside of the cell, which are in higher concentrations. When I took biology at the University of Victoria, my teacher described this setup as a banana in the ocean. So a banana full of potassium floating in a salty ocean. So the potassium on the inside creates this concentration gradient, and the sodium and chloride on the outside create this concentration gradient, and their negative and positive charges also create electrical gradients. At rest, this is how the ions are distributed. As we saw, the potassium and the protein anions are more plentiful on the inside, and the sodium and chloride are more plentiful on the outside. Because the membrane doesn't allow these large proteins through, the negative charge proteins are just in the inside of the cell. So the inside of the cells kind of starts off with this large negative in its column. So potassium and chloride channels are open usually in the cells, and these allow potassium and chloride to pass freely through. But sodium channels have gates, and generally sodium is kept out of the cell by these gates. And then finally, sodium and potassium are pumped in and out of the cell respectively. So sodium is pumped into the cell, and sodium is pumped out of the cell, and potassium is pumped into the cell to make it more like a banana in the ocean. And these are actively done to keep up these concentration gradients. So here we can see just a summary of what I was just talking about. Potassium can enter and leave the cell freely. It will be inclined to leave the cell because of the concentration gradient, but also to enter the cell because of all the negativity on the inside of the cell. Sodium wants to come in because of the negativity and because of how much less sodium is on the inside, but it can't because of the gate. And because sodium wants to come in so bad, you have to actively pump it out when you want to recreate the resting membrane potential. So you can use these pumps to pump in 2 potassium and pump out 3 sodium to recreate the potential. So the anions are manufactured in the cell, as we saw last week. And because they can't leave the cell, they create this large negativity at rest. They also accumulate these potassium ions so that there are about 20 times more of them inside than outside the cell. So they want to be drawn out of the cell because of the concentration gradient, as we mentioned. Sodium ions are much more plentiful outside, about 10 times as many outside than inside. And so they want the sodium wants to come into the cell. And this contributes to the increase in positivity outside compared to inside. But mostly sodium can't come in because these gates are always blocking it until they open up. Finally, chloride ions move in and out of the cell freely through the channels in the membrane like potassium. But at equilibrium, when the concentration and the voltage gradient are equal, is the same. The equilibrium point of chloride, when their concentration and voltage gradient are equal, is the same as the membrane's resting potential. So when the membrane is at rest, chloride is generally happy being where it is on the inside and the outside. So not much of the resting potential is due to this chloride because it's free to move. So because the large anions can't move, it's negative on the inside. And potassium and chloride move into and out of the cell freely, but the sodium stays out, and sodium and potassium get pumped in and out, as we saw. This is a slide we already saw, just to refresh. So in sum, the inside is more negatively charged than the outside, just like a battery, and this creates this potential energy for when the gates open for things to change very quickly. So if any of the ions concentrations change between the inside and the outside of the membrane, then the membrane voltage is going to change. If a bunch of sodium rushes in, then it will become more negative on the inside. If a bunch of chloride goes out, it will become more negative on the, or which charge is chloride. If a bunch of chloride goes out, it'll become more positive on the inside. So we call these graded potentials. They're small fluctuations in voltage across the cell membrane caused by input from other neurons. And these can occur in two types: hyperpolarization. So when the electrical charge across the membrane increases. So for instance, when chloride ions come in or potassium ions go out. Or depolarization, which is something that occurs prior to an action potential. Which is a decrease in the electrical charge, so less negative inside and compared to the outside. So instead of -70, it will approach -50 or 40 or 30. There'll be less potential difference between the two. And this is usually because of sodium coming into the cell and making it more positive. So for the membrane to become hyperpolarized, the outside of its cell must become more positive, which can happen if potassium leaves the cell, or it could become hyperpolarized by chloride coming into the cell. And when it becomes hyperpolarized, it's going to be less likely to transmit information onto the next neurons, as we'll see. Conversely, depolarization occurs when the sodium gates open up and these positive sodium ions rush into the cell, creating an increase in positivity towards 0 from -70. So when these depolarizations occur to a large enough extent, they can trigger something called an action potential, which the cell uses to transmit information onto the next cells. So an action potential is a large and brief change in the polarity of an axon from negative on the inside to positive on the outside, flipping it and then back to normal. It lasts about a millisecond. And it doesn't occur until the particular membrane in which it's going to occur has reached a certain threshold. So there's a certain voltage that the neuron becomes depolarized to, at which time it will fire one of these action potentials. And when this happens is when those gates that we saw, the sodium and also potassium gates, open up or channels open up due to the change in voltage. So the actual change in membrane voltage opens the channels and creates this cascade effect where they then get depolarized more and more and more, creating the action potential. So when the neuron depolarizes to about -40 is when this process might start. Okay, so here we can see the cell becoming depolarized, passing the threshold and creating an action potential. It takes about a millisecond to complete. here, as we zoom out, we can see how many action potentials any one given neuron might create, even in some small time period like 40 milliseconds. We can see that the action potential involves a change from negative to positive between inside and outside of the cell, and then back to very negative, and then resetting to the baseline resting membrane potential. The most important part of this action potential is these voltage sensitive channels that are embedded in the membrane that only open once a certain voltage has reached. So all this depolarizing input is coming into the cell, and the cell is taking all this input and it's making it more and more and more depolarized, depolarized, depolarized. At a certain threshold, the channels, these voltage sensitive channels, will open up and a bunch more sodium and potassium will rush through them and knock off this process that creates the action potential. So at the resting membrane potential, therefore, these voltage sensitive channels are closed, and the sodium channels open up first and the potassium open up a little bit later. That is, the sodium channels are opening up at a lower, a more negative voltage than the potassium. Okay, so when a minus 40 millivolt resting membrane potential is reached, the sodium and potassium gates can open. Sodium rushes in, potassium rushes out. As the sodium rushes in, the first thing that happens is when this major depolarization occurs. And then sodium and potassium both start to occur. And once the sodium stops, potassium really starts to leave, and that creates this over negativity on the inside. And then the sodium potassium pumps start kicking in and recalibrating the balance, and it gets back to about minus 70. So when a large concentration of first sodium and then potassium crosses the membrane rapidly, first it gets depolarized by sodium. You can use the drug tetrodotoxin, which is found in pufferfishes, which blocks sodium channels and stops this depolarization from occurring, which is why it's a deadly neurotoxin. And the action potentials that occur are different because they're entirely due to just the potassium effect. You can conversely do the opposite, where you just block the potassium channels and you see a smaller action potential just from the sodium itself. This is the way that the scientists identified the relative contributions of sodium and potassium to the action potential. Right, so you can block the potassium channel and see just the sodium influx, or you can block the sodium channel and you see just the potassium efflux. You can see their relative time with respect to one another. I think we've already covered this, that once they reach this threshold voltage, they both open up and allow their ions to flow. And as we mentioned, the sodium actually opened before the potassium channels. And so the change due to sodium happens before the change due to potassium. And the sodium channel actually has two gates. And so actually the first event that pushes the depolarization into this first spike is only when both gates are open, and then the first gate quickly closes at about minus 30 millivolts. And potassium open more slowly, they remain open longer. And they even counteract the depolarization by hyperpolarizing the neuron for a brief period of time. We talked about these voltage sensitive channels not allowing any potassium or sodium through unless they get activated by particular resting membrane voltage. Now, because of the need to recreate the voltage and concentration gradients before these things can happen again, the neuron has something called a refractory period before it can fire another action potential. When the neuron is becoming repolarized after the sodium influx, it can't fire an action potential at all, because the second sodium gate, the one that isn't voltage sensitive is closed during this time, and so no more sodium could come in, even though there might be a concentration gradient. And later on, the relative refractory period, you need more electrical current to produce an action potential, so the neuron is a little bit tired out while it recalibrates its potential, because the potassium channels are still open, and so there's not as much of a concentration or voltage gradient. So here is when at rest, just the second gate of sodium is open. Once the cell becomes depolarized, the first gate opens, sodium rushes into the cell and depolarizes it. This is the absolute refractory period, which continues on as the potassium gate opens up. Once the first sodium gate closes, we enter what's called the relative refractory period, in which one can fire an action potential by stimulating the neuron, but needs to stimulate it more than normal. And then finally, a return to resting membrane potential. And think about this like the flushing of a toilet. So during the original flush is the absolute refractory period, and you can't press the flush again. And then while the tank's being refilled, the toilet's in this relative refractory period. You can flush, but it's not as good of a flush. So we call the nerve impulse the action potential moving down the axon. So the action potential actually has to be transmitted down this long axon, which we even learned was very long in some of cases for our brainstem down to our big toe. So how does this information travel all the way down this long axon? Because of the refractory period, the nerve signal can really only travel in one direction, because it can't come back, because the parts of the membrane that had just fired can't fire again for a while. And also, as it moves down the axon, it seems to be the same at each place of the axon. So if you stimulate the head of an axon, you can watch one of these action potentials move down through the axon. You can see that it can't move back the other way because of the refractory period of those cells. However, the voltage influx from the sodium and potassium can affect the membrane sensitive, the voltage sensitive channels at nearby parts of the membrane, creating this cascading effect that lets the signal move down the length of the axon. One can think about this also like dominoes. So each domino knocks down the next one, and each one is a single unit and can't be a bigger or smaller fall. And it also can't go back the other way because the previous dominoes have already lost their potential energy. So the refractory period prevents signals from moving back up the axon the other direction. Now, these axons, as we saw, are quite long, and to travel down the long axon from my brainstem to my big toe, or even in the giraffe we saw at the zoo last week in Calgary, from the giraffe's brainstem down to its big toe of its back foot, the information would go very slow given these thin axons. To make up for this, the nervous system has designed evolution has designed myelin to wrap around these axons to increase the speed by which they can conduct information. We saw that myelin is produced by the oligodendria and the Schwann cells, respectively, in the CNS and PNS, and that the myelin speeds up the transmission of information along the axon. It does this by creating these small windows along the axon in which there's no myelin, called nodes of Ranvier. And these tiny gaps enable the voltage sensitive channels to open up every so often in the axon, enabling something called saltatory conduction or jumping conduction. So here we can see the oligodendria and Schwann cells wrapped around those axons, and the gaps between them called the nodes of Ranvier. So we talked about saltatory conduction, or the dancing between each of these successive nodes. Right, so as the sodium comes in and potassium leaves, that change in current flows over to the next node of Ranvier and opens the voltage sensitive channels in the next node, transmitting this wave down the axon. And it's unable to go back the other way because of the refractory period. If this myelin becomes damaged throughout our life or in disease, then the neuron has trouble transmitting information down the axons. And multiple sclerosis, or MS, is a degradation of the myelin formed by oligodendrocytes in the central nervous system. And it disrupts the functioning of our neurons and leads to transmission problems out to our muscles of our body as well. So next we'll talk about something we've already alluded to when we talked about graded potentials, in that neurons have to integrate information together from thousands of other neurons before they can decide whether to fire an action potential or not. So one neuron can connect to up to 50,000 other preceding neurons that send it information. So that receiving neuron might get all sorts of inhibitory and excitatory signals from all these different neurons. And the cell body is kind of a junction between the dendritic tree that picks up all these inputs and the axon, which can send outputs. And so the cell body has to essentially take a vote of all the input to decide if it's going to be depolarized enough to open up the voltage sensitive channels at the axon. So we talked about these excitatory and inhibitory graded potentials, and we can call them excitatory postsynaptic potentials. So a brief depolarization of a membrane in response to stimulation. And now that that neuron is more depolarized, it's more likely to fire an action potential. So instead of a small electrical stimulation, you can think of the preceding neuron stimulating that neuron and making it more likely to fire the action potential. You can also have an inhibitory postsynaptic potential, which is the opposite of brief hyperpolarization in response to stimulation, which makes it less likely to fire an action potential. These EPSPs and IPSPs from the incoming inputs to the neuron are each only lasting a few milliseconds, and they decay, and the resting membrane potential is restored. So they have to be combined quick enough, and many of them, in order to reach the threshold to fire an action potential. EPSPs are usually associated with opening of sodium channels, allowing sodium to rush in and depolarize the cell, where IPSPs open up potassium or chloride channels and do the opposite and create a hyperpolarization. So excitatory sensory pathways coming into a motor neuron. If you measure from the resting membrane, if you measure from the membrane as they come in, you can see this change in voltage to depolarize the neuron as you stimulate the preceding neuron to get it to input into this neuron. If you stimulate an inhibitory incoming neuron, however, you can create a hyperpolarization, making the neuron less likely to fire an action potential. So we can see that we either stimulate the incoming neurons and then record from the input motor neuron. And that neuron can either be excited or inhibited from these incoming neurons. This was another set of research that went on to win the Nobel Prize by a guy named Eccles, who identified this inhibitory and excitatory inputs to neurons by using this stimulation and recording techniques. These EPSPs and IPSPs need to sum together in order to create a large enough depolarization to make the cell fire. And they can sum together in time and in space, and in time and space at the same time at once. So temporal summation means that the pulses, the incoming EPSPs and IPSPs might come close together in time. Spatial summation means they might come close together on the dendrite of that incoming neuron. Okay, so here's EPSPs and IPSPs when they're spread out in time, but you can see if they come into the neuron close enough in time, they create this synergistic effect and create a large enough change in the resting membrane potential that they push it towards depolarization. in relation to this temporal summation, if these two incoming neurons that are close in time occur far away from each other on the dendritic tree, they're going to have less of a summation effect than if they occur very close together on the dendritic tree. So there's spatial summation as well, and the closer together two inputs are, the more they'll contribute to a depolarization. So the neuron sums up all these inputs that are close in time and space, and it provides some output of how much all those influences all vote towards it firing or not firing. And if that summed ionic input exceeds this threshold, if they depolarize it enough to get past minus 50 millivolts, once it gets to the axon hillock, the start of the axon, then it will initiate an action potential. And you can think of sodium's role in this summation because the sodium kind of adds together if they're occurring close enough together in time and space. So the more sodium you can get in that area close in time and space, the closer it can get to the threshold membrane potential. And similar with potassium. Finally, the axon hillock, this boundary between the cell body and the axon, where something called the initial segment of the axon is the junction between the cell body and the axon. It has tons of these voltage sensitive channels. So this is where all the voltage sensitive channels are that make the vote from all the dendrites, whether they've depolarized the cell enough. So they're integrated together as they reach the axon hillock. And if enough EPSPs balance out the IPSPs, they don't become depolarized enough to begin the action potential. So if all these incoming IPSPs and EPSPs combine to depolarize the cell at this initial segment enough so that it passes the threshold, it will create this action potential. And the action potential can then travel down the axon towards the next neurons. So because the cell body itself doesn't have any voltage channels, the neuron and the dendrites don't create action potentials. However, there are some cases where there's some voltage sensitive gates on dendrites. And this could serve a role in something that might be called back propagation, or the movement of information from one neuron back to the previous neurons which influenced it. So this could send an action potential from the axon hillock up into the dendrites, and these could signal to the dendrites that that neuron is currently sending out an action potential, and it might help play a role in the plasticity of neurons in learning by telling previous neurons whether their vote counted or not, just like the feedback we all got from the election yesterday. So just another, some peculiarities of some neurons. Some species don't have any dendritic branches at all. Some ion channels don't respond to voltages, they respond to light in certain animals. And this has allowed scientists to do a cool techniques called optogenetics, which we'll talk about on the next slide. And all these different types of neurons that have evolved indicate how much our nervous system can capitalize on modifications of these neurons. It can create neurons to do certain things in certain situations given adaptive and reproductive pressures. Optogenetics is an example of that, how neurons in these two types of animals have developed really interesting adaptations in which they are activated or they change their resting membrane potential by the influx of light. So here, blue light opens up a sodium-potassium channel, or green-yellow light can open up this chloride pump and inhibit the cell. Optogenetics involves the infection of an animal with a virus that also includes the genetic fingerprints for these channels so that they can be expressed on the neurons of that animal, at which time then the scientist can shine this particular color light on the animal's brain and open up those channels experimentally when they want, allowing very tight control over excitation and inhibition of the neurons. So finally, we'll talk about the input and the output of the nervous system and how electrical signals play a role in these two parts of the communicative process of neurons as well. So when we pick up sensory information about the world, which will be the topic of our second midterm generally, we receive information from our body, our ears, our eyes, and also our chemical receptors in our nose and our mouth, as well as balance receptors in our inner ear. So all these neurons attached to all these receptors have ion channels in their membranes, and generally these ion channels initiate a cascade of events that leads to nerve signals being sent out of the axons in different ways depending on the type of incoming information. So touch is one example. So we can identify individual hairs on our body. Our arm is a good one because we have increased sensitivity there, and you can move individual hairs and you can feel slight displacements of these individual hairs of your body. So each hair has the dendrites of a neuron wrapped around it, and as the hair displaces, as you move the hair side to side, those channels get stretched and opened up, physically, mechanically opened. And this opening allows sodium to rush in and depolarize the cell and then send signals into your body to indicate that you were getting tickled at that location. So these mechanical changes in the hair follicle open up these sodium channels and create a depolarization, producing a nerve impulse that gets sent down the axon. On the output side, spinal motor neurons send output to our muscles, and the axons of each motor neuron synapse onto the target muscle. And when the signal gets sent to the muscle, they let the muscle contract. And the axon attaches to the muscle at an area called the end plate. So it's part of the muscle membrane that's contacted by the axon. The axon releases a chemical called acetylcholine. Just like we'll see next week, many of these axons, when they send the signal to the next neuron, which creates the IPSPs and EPSPs, they do this by releasing a chemical. And in the case of muscle cells, that chemical is acetylcholine, which gets released onto the end plate of the muscle. So this acetylcholine attaches to transmitters, so acetylcholine sensitive channels on the muscle, and those channels open and allow sodium and potassium into the muscle membrane, depolarizing the muscle. Then these muscles create action potentials, and it's actually the action potentials that contract the muscles. So here's the incoming axon from the motor nerve, releasing acetylcholine onto the end plate at the axon terminal. Acetylcholine can bind to these receptors, opening up channels, letting in sodium and letting out potassium. And these open up nearby voltage sensitive channels, creating this cascading effect, allowing the whole muscle to contract at once. So that leads us to the end of this chapter 4. We're going to fill in the final details in chapter 5 about how this communicative process works by understanding in the brain the chemical signals that get transmitted from these axons to the receiving dendrites of the next neuron are going to be neurotransmitters being sent through the synapse. So next week we'll learn about the synapse and about neurotransmitter systems as well, which will bring us to the end of the material for this first midterm. So what we covered today is the electrical activity of the nervous system, first with the membrane, and then how neurons can integrate information from many neurons together and use this resting membrane potential as a kind of vote tally to decide if they should send out a signal to the next neuron. We talked about how incoming sensory information from the world is transduced. It's called transduction, is the process of turning this external sensory information into electrical activity in our nervous system, the topic of our second midterm. And we learned about how this transduction happens in our sensory receptors, and then we learned how the motor output gets sent out to our muscles, activating them to move about the world. Have a great week.